So we can begin with the session, I think. Um, yeah, I am audible. Okay, so those of who are joining for the first time, although there are recordings for these sessions that are available, so I'm sure you might have gone through some recording if not joined before. But let me just introduce myself again. So I'm Arveen Kaur. I'm a PhD scholar at Department of Biological Sciences and Bioengineering at IIT Kanpur. And I am the TA for this course that is Neurobiology and that is being taken by Professor Nitin Gupta. Right. And this is actually an extra session I did in form of an extra session when we had our last discussion session, which was the week 4 discussion session, right? But over there, one thing that I was not sure of is when that session would be, because those that had not been intimated to us by then, right? So anyway, now we are having that session, although it has been a little time since we last were looking at this course. Those of who would have followed those videos or are following this revision session might remember that even in the week 4 session, we were unable to actually complete the revision of the entire syllabus, right? We had to begin somewhere near tuning of the wavelength for auditory and ophthalmological signals, right? Visual and auditory signals. So that is what we are going to pick up today. Along with that, if you guys have any kind of doubts, then now is the time. I went through the discussion forum. There was one prevailing doubt that was present that also I will solve, which is as to why the inside of the membrane is less is more negative, although there is abundance of potassium ions, which is a positive ion inside. Right. So there are multiple, mechanisms at play over there. So I will try to take up that also. Before moving to that particular question, I'll just finish the revision of the part that was remaining, right? And along with that, if you guys have any queries, you may ask me. Right. In the previous sessions, we have also actually, okay, yeah. In the previous sessions, we have also actually been able to go through all these questions, previous year questions before we actually switched to the revision. But that was for 4 weeks, I had your previous year questions as well. Now I no longer have, access to the previous year's examination. So I hope that, okay, there are some people in the lobby, I think. Just a moment. Yeah, I think it will not hamper my presentation. Just let me check. No, I'll just wait. I'll stop sharing and let's share again, just so that there's no confusion at all, right? So that I'll also know that.
चलो क्लास शुरू हो गया क्लास शुरू हो गया you will go a long way if you can do that.
Okay. So as I was saying, I did not really have access to your previous year's question paper. So the questions that we have dealt with in the class, this revision lectures as well. Is somebody trying to say something? Okay, I think that was my mistake. Yeah. So those and the ones that you have done in your assessment this year, they should be your model questions, right, for the exam if any of you have registered for the exams. Other than that, the portion that was left in week 4 and that module 4 and 5 lectures that should have been covered in week 4. But due to lack of time, we were unable to do that. We'll pick that up right now, right? Okay. So this was actually the last slide that we did discuss, or the last piece of information that we did discuss in week four's module. And this basically talked about sensory modalities, right? What are the different kinds of sensory modalities? What are the kind of stimulus that they detect? What is the receptor, the kind of receptor that is taking in that signal? And what are the cells that are involved, right? So over here, there were some key things that we learned that apart from the photoreceptor that we do know that there are photoreceptors in the eyes. Apart from these, and the chemoreceptors, which are the two commonly known receptors, right? Most of the people know that, okay, these are the kinds of receptors that are present, right? Over here, we also learned that, yeah, there are a lot of receptors that are actually mechanically activated, right? So they are mechanoreceptors, whether they are the hair in the cochlea or, the movement of the skin cells on action of or on touch, on some kind of touch, right? These there are multiple kinds of mechanoreceptors that are present, which are mechanically activated, right? Thereafter, there was another class that we dealt with, which is the thermoreceptor, right? So this thermoreceptor is basically for proprioception or for pain sensation from outside, right? So it will judge, or it might have channels that might open or close in response to some temperature changes, right? So there are thermoreceptors as well. So this was the last slide we dealt with. After this, there was a very, very interesting topic that we couldn't cover last time, which is basically tuning of different kinds of sensory modalities, right? So first, if we talk about tuning of an auditory receptor, right? So this is a little simpler, and then we will also move to the visual receptor, right? That is a little more, that will require a little more explanation to understand. So when we talk about tuning of an auditory receptor, what is an auditory receptor? It is basically a receptor that is going to perceive any sound or any frequency changes that are around us so that it can convert that frequency change into a sound that we can perceive, right? Now we know that humans are susceptible or can hear sound within a certain range, right? So if it is a very high frequency, we won't be able to hear it. If it is a very low frequency, we will not be able to hear it, right? For example, the ultrasonic sounds, we can't hear it, but there are other mammals such as bats that can actually hear it, right? So for us, what is the frequency range that we can hear? Does anybody know that? So it is basically 2 to 20,000 hertz, right? Or 2 to 2 kilohertz, right? So this is the frequency range that is audible to us. Now, within this range, also there are multiple different kinds of receptors that are responsible for different kinds of frequency response, right? So that are responsible for aiding our hearing to a certain kind of frequency. Right. So, for example, I will have a bunch of receptors that will be responsible for me being able to hear, say, 5 kilohertz of frequency, right? There will be a bunch of receptors that will be responsible for me being able to listen to 20 kilohertz of frequency, right? And there will be some different kinds of receptors. These receptors will also probably be in the bat, right? Because we are evolutionarily related. But apart from that, they might have different kinds of receptors as well that we lack, that basically aid them to hear the ultrasonic sound waves as well, right? So what does this mean? That it is not like there is a bunch of receptors that is present on your pinna or on your inner ear canal that can basically detect any frequency, right? There are multiple heterogeneous receptors that are present, and some of those receptors are responsible for some particular frequency, right? For hearing some particular frequency. For example, in this graph, you can see that the y-axis talks about sound intensity in decibel, and the x-axis talks about sound frequency in kilohertz, right? So there is a receptor that is basically tuned to listen to 2 kilohertz of frequency, right? So that receptor, even at a very low noise, right, a very low sound, right, what is decibel or what is sound intensity is how high the volume is, right? So it is kind of like uh, maybe even at 2 hertz, if there is a whisper, this particular receptor will be able to hear, right? Even at the level of a whisper, right? It does not really need somebody to shout at 2 hertz frequency so that this particular receptor can be activated. As long as the frequency that is coming out of that person, like when the person is shouting, is close to 2 kilohertz, right? So that is the only prerequisite for this particular receptor, right? That is what this particular graph shows, that it will probably be able to hear some sound from 1 kilohertz as well. But the sound intensity required for the person to be able to hear 1 kilohertz of sound using the same receptor that is tuned to 2 kilohertz is very, very high, right? So you can also relate it to the radios. I don't know how many of you, I think this generation might have heard. I'm not sure about the upcoming one. Right. So I think you will be available aware about the radios or the FM channels, frequency modulated channels in the car, right? That is the automatic radio that is playing, right? If you are not plugging in your own list of songs through your phone or through a USB, that is what you can hear when you are changing those channels, right? So, for example, at 101, there is a particular channel named X, right? And at 100.5, there is another channel named B, right? So, if, for example, you tune the knob to 101, you will hear this channel A. But if, for example, you tune it to 100.9, right, you might be able to hear a little bit of 100.5 and a little bit of 101, right? Similarly, if you tune it exactly to 100.5, it will give you the exact sound that's coming from that one channel, right? But a little above, even 100.6 or 100.4, it will be able to maybe capture some other sounds also, right? So at this particular frequency, it is very, very specific to this, right? But a little, like it will have a certain range that it can detect, just that you need to increase the sound frequency, sound intensity over there, right? So that is how. So that is basically how what is known as tuning of an auditory receptor. So our auditory receptors, our visual receptors, all are tuned like you tune your instruments. All of these are also tuned to a particular frequency or to a particular intensity of light. So different receptors within a modality may respond to different ranges of the stimuli, right? So for example, I have a small area in my inner ear and it has 10 different receptors, right? And out of these 10 different receptors, three of them are 2 kilohertz frequency receptors. Two of them are tuned to 5 kilohertz frequency. One of them is tuned to zero point 0.5 kilohertz frequency, and so on. Right. So this means that within that little area also, there are multiple kinds of sound frequency that can be very distinguishably identified. Right. So that is what this means. Right. So that was tuning curves for auditory receptors, right? Now, when we talk about tuning for photoreceptors, it basically means that we are talking about something that is related to visual aid, right? When we talk about this visual aid, what happens is we have already studied in the previous in your lectures with sir also you have studied, and in the previous revisions lecture also you revised again that there are these three kinds of cones that are present. One is the blue cone, which peaks basically between this purple blue horizon, right? There is a green cone which peaks at this green horizon, and there's a red cone that peaks at this yellow red horizon, right? And these you can see have a specific wavelength in which they function, right? So that is one thing. And then we also saw that there are rods, right? So cones are the cells that are active during daytime generally, or when the light intensity is good enough, right? But when during night there is very little light, it is not like you cannot differentiate objects or you can't differentiate colors, right? That still you can do that, right? That is because of the presence of these cells, which are rods, right? Although they peak at this blue green wavelength, they spread across a lot more area or across a lot more wavelength than do any of the individual cones, right? So that is how they aid us to visualize at night, and only one kinds of rods are present, right? With respect to multiple kinds of cones, we just have one kind of rod because it is present across a wider spectrum, right? So it can handle most of the wavelengths of lights that we see at night, right? So that was one thing, right? Now these are also tuned, right? So that is why this blue one is giving the like it is peaking at this 437 nanometer. The rods are giving a peak at 498 nanometers. So this means that these photoreceptors are also tuned, right? That they will peak at this point, and the green, for example, will almost, detect wavelengths from 450 to around 650 nanometer, but it will peak somewhere around 530 nanometer, right? Similar for the red cone, it will basically from around 475 to around 675 it can detect, but it will peak at around 564 nanometer, right? So, yeah. So this means that these rods and cones are also actually tuned, right? So this that is how we actually have gotten this kind of wavelength, right? And this is the reason that there are different combinations of colors that are actually available, right? So coding of a color by combination of differently tuned receptors is how we can visualize so many colors, right? We can distinguish between two kinds of blues. We can distinguish between maybe multiple types of greens, right? There are so many greens. Every tree that you see has a different shade of green, right? Even within the tree, there are so many shades of greens that are present. And it is not like it looks uniform to you, right? It is only green, but it doesn't look uniform to you, right? You can distinguish between this dark green, light green, bottle green, olive green, etc., right? So there are multiple distinguishable features in colors. Now, how does that arise is that there are multiple different kinds of photoreceptors that are going off, right? So, for example, if I am looking at something that is a little, greenish yellow or yellowish red, right? Let us say this arrow was going through this angle, right? So that it was greenish yellow. So that means that may some of the photoreceptors will be activated in the green cones and some of the photoreceptors will be activated in the red cone, right? Now, what is the percentage of photoreceptors of green cones versus percentage of photoreceptors of the red cone that are actually activated will determine like which color I am seeing, right? Am I seeing greenish yellow or I'm seeing dark yellow or I'm seeing mustard, it will depend on what is the ratio of green cones to red cones that are being activated over here, right? So that can very well be explained by such a form of prism, right? And that is how even those like these days there are these smart bulbs and all that are coming up, and they advertise that they can have 6 million or 60 million something colors you can pick in them, right? They can light up in any color that you pick up on that app, right? Now, how is that possible? Because your eyes should be able to distinguish also between 6 million different colors, right? And they are, because there are n number of possibilities when we look at such a graph, right? So, the tuning of individual receptors basically uh, helps in, you know, them coming up in different combinations also later on, so that you can distinguish different variations in certain colors. So that is basically why a visual tuning of receptors is also a thing and it is required, right? Then we also talk about receptive fields. So when we are talking about visual stimuli, we need to understand that it is not only the colors that we are observing, right? It is also you look at something, I'm looking at my laptop, I can make out that what is the distance of my laptop to from me, right? If I'm looking at my mouse, for instance, I can make out how far away it is kept, so that I can at one go only I can extend my hand and I can reach to it, right? And then I can maybe point this laser towards receptive field, or I can maybe exit this presentation, end this meeting, whatever, right? So I have some kind of visual sense, right? Just by, spatial sense, just by visually looking at certain objects, right? So that is the kind of power that your visual reception has. So there is a lot of spatial information that you get, right? What is the kind of spatial information that you get? The location of the stimulus, for instance, right? For example, if I get a pop-up that okay, somebody is trying to join in this meeting, right? So I get a visual stimulus, right? After that, I do, admit that person to this meeting. So that is how I know, right? That okay, there is this pop-up, it is at this place. I know where my mouse is. I know how to, handle it. And that is how I can admit somebody to a meeting, right? Then you can also understand the size and shape of objects, right? So I know that I have a rectangular screen in front of me, right? Outside, I have multiple, plants. They have a very different kind of morphology, much different than simple circles and squares. Then you can also look at the fine details within the shape, right? So one kind of fine detail could be that I am able to read what is written on the screen, right? It is not just me looking at a red block and a black block. No, right? I can read that because that is the detail that it is given, right? Outside, if I look at the same plant, I can see that okay, it has multiple branches. There are some buds that are present on that plant, right? Maybe it will bloom in some time, right? I cannot currently make out the color of the buds, right? So there are different kinds of information that you get just by looking at at an object, right? Then these spatial abilities are linked to the receptive field of each neuron, each sensory neuron, the region of space where stimulation excites the cell, right? We can also define receptive fields for neuron in subsequent layers of cell. So, this is what I can see on a bigger, this is the bigger picture of what I can see, right? But do you think that my entire screen is an outcome of one particular photoreceptor or one particular sensory neuron? I feel that there will be multiple sensory neurons or photoreceptors that will be activated when I look at the screen, right? So that multiple of them might send some information to the optic nerve, which carries it forward to the brain that is actually processing it, right? So, what are the kinds of or what is the region of neurons that is being stimulated is actually my receptive field, right? So, for example, if I'm looking over here, I do not have very good vision of what is happening on my side, right? Maybe if something moves on the side, I might get alerted because there is some kind of peripheral vision. A little maybe uh, because a lot of neurons are active in the middle region of my eye, and only a few neurons are active on the side of my eye when I'm looking towards the screen, right? So that basically means that the receptive field is focused in the front, but yeah, I have some reception towards the side as well, because maybe some 4 or 5 neurons might be activated over there as well, right? So why is that important? Obviously, because right now my motivation or my primary goal is to actually look at this presentation and explain this presentation, right? But why are my peripheral ones active then? Because evolutionarily, there, like, I should be aware of my surroundings, right? That is what is my peripheral vision is what is making me aware of my surroundings, right? If I could only look ahead and I would not know if something is coming ahead from the side, that something could be potentially dangerous, right? How would I save myself from, right? So, yeah. Now, this is again a representation of the homunculus, essentially homunculus or the somatosensory homunculus in the right cerebral hemisphere. And over here also you can see that the visual, which is the eye, I can't see it written over here. Yeah, the eye. All muscles are the muscles and whatever helps us in manipulation, such as our fingers, the tongue, because it is continuously moving. It helps us in talking also. It helps us in our taste also, right? And our lips, that are the muscles that are involved in these movements. These are the ones that have the most space dedicated to them in the somatosensory cortex. But apart from that, visual stimuli is also one of the major ones, right? So eye also plays a has a very significant role over there, right? So this is just a representation of that. Now, let us also, from a different kind of example, understand what receptive fields are. So, what did we understand it based on? What kind of what receptive fields could mean with respect to visual stimulus, right? Now, there are other kinds of stimulus also that are present, right? Receptive fields of sensory neurons in the somatosensory and visual systems define the spatial resolution of a stimulus, right? So, for example, what will happen is that maybe I am feeling that I have some kind of pain in my hand, right? Once I look at my hand, it could be a joint pain. Maybe I won't be able to make out where in the joint it is paining, right? But maybe if I look at it, I might find that, okay, my hand is bruised over here, right? Or I might find that, okay, there is some kind of a red spot over here, right? There's some kind of an inflammation or vasodilation over here. So, by the aid of both what I'm feeling, right, the sensory neurons from that region that are taking the this stimulus, sorry, let me just gather myself that are taking the information about the stimulus to my brain will also direct it to that place, that okay, that is how I know without looking also that it is not in my finger, it is in my wrist, right? And once I look at my wrist, or it might be a very localized pain, so without like looking also I can say that, okay, I think that it is paining, it is really paining over here, maybe I should look what has happened, right? So it is what is a receptive field, right? So if for example somebody was pressing my hand, or if my hand was caught between a door, right, the receptive field would be larger, right, because my entire hand would hurt. Versus if somebody was pricking me with a needle, right, or somebody came and pinched me at some point, right, so the receptive field is shorter, right? Only maybe this neuron will be activated and it will tell me that, okay, something is pricking me, or something is maybe somebody is kind of pinching me. But if the entire receptive field is activated, maybe I will know, okay, I think there is some kind of an injury in my hand. Maybe my hand is caught between something, right? Maybe you have your hand in hot water, and that is why your entire hand is giving out signals, right? All of these could be considered as being activated and being in this red color. So your hand, your brain at that time, even if it looks at it, will not be able to make out which part is more burnt, right? Because your entire hand was in hot boiling water, right? So even if you take out, there will be a localized sensation of pain, right? Sorry, a uniform global sensation of pain. It will not be localized to a particular region, or you will not be able to localize it rather, because there is so much neurons that are firing that it will be hard for you to localize which point has burned the most, right? So that is basically another example of a receptive field, right? Now, when we talk with respect to pixels, right? Uh, a dense population of receptors within a small receptive field lead to finer resolution of spatial details, right? So, for example, this is a fabulous example that sir had given. That, for example, you have a box, a square box. Say that it is a 1 by 1 dimensional box, right? In this square box, in this 1 by 1 dimension, say you have 400 receptors, right? If you have 400 receptors, then each receptor will cover a larger area, right? And this is a kind of spatial information that you will get. So the receptive field for all this, for a single, receptor will be larger, right? So this is the kind of information that you will get. Now, if in the same area, 1 by 1 area, you increase the density of the receptors, right? So, meaning that I still have one pizza box, but inside that one pizza box, now instead of one pizza, I have put two pizzas, right? So I have not increased the number of slices of a pizza, maybe, right? But I have put two pizzas, right? It is possible there's a lot of space over there, right? So that means the density has gone up within that pizza box, right? So earlier, if I had a pizza box with one pizza and it was able to feed, say, two people, now I have doubled the amount of pizza that is present in that same box, and now it can, feed eight people maybe, right? Or four people if it is just double, right? So what does that mean? That it has occupied the same, in the same space, I have been able to feed more people. Similarly, in the same space over here, 1 by 1, there are more number of receptors that are present. So each receptor will have lesser space that is present within it, right? So if that one pizza was sitting, it might have been fluffed up and sitting in a box alone. But if I crammed another pizza into the box, the individual space for the first pizza might have been lesser, right? Or for both the pizzas might have been lesser, the individual space. But the kind of function that they had to perform is actually better, right? Because now instead of two, they can feed four people, right? So similarly, if you keep on increasing the density of the receptors, since one receptor has to perform lesser function, right? For example, to begin with, we had eight people and we had one pizza, right? So that means that one piece of a pizza should be able to feed one person sufficiently, right? But when we say that we have eight people and we have four pizzas that are crammed up into that same box, which means that now one piece is no longer required to completely fulfill one person, right? One person can maybe have up to four pieces, right, to fulfill their or satiate their hunger, right? So now one piece is not responsible for satiating somebody's hunger. Now we have four pieces to satiate that person's hunger, right? And that is the case that we get over here also when we say that there are a high density of receptors that are actually present, right? Okay, so the okay, we'll come to this slide. Just let me have a water break for a moment. Yeah. Visible. I'll just check the layout once again. Yeah. Yeah. So now that we have talked about the stimulus and how it is, how the density of the receptors will define like what is the kind of resolution that you are getting out of one single stimulus, right? Now we also should see what is stimulus intensity and how basically it will be affected, right? So stimulus intensity is basically how intense a signal we are getting, be it a visual stimulus or a sensory stimulus or sorry, touch stimulus or any other kind of olfactory stimulus, any such stimulus, right? It is encoded by frequency of action potentials, right? So for example, somebody barely touched me at one point on my wrist, right? So that has sent some signal to my brain and it says that, okay, there was some sensation that I felt over here, right? Versus that there was somebody who was basically continuously, frequently doing this, right? Then maybe the stimulus, the neurons that are taking the stimulus of this touch on this part of the wrist is firing again and again, right? So there are multiple action potentials that are being built up, right? And it is firing again and again. So my brain will know that, okay, the intensity of the stimulus is higher, so I might be alerted that, okay, I should pay attention to something that is happening over here. Say that you have a toddler, right? And you are busy doing some work and that toddler just came and touched you over here, right? That toddler wants your attention. So he or she just came and touched you over here, right? And you are just working and you are deeply engrossed in your work and you don't pay attention. But if the toddler keeps repeating it, your brain will know that, okay, it is something that is important and okay, I should pay heed to that stimulus, right? And then your attention might go on to that person even though you have very important work, right? So you might then realize that, okay, somebody was actually trying to call me. Okay, what is your what is the thing that you need, or how should I help you out, right? So that is basically why repeated action potentials or stimulus intensity is also considered, right? Now there was, like, it is not given over here, but like for example, there was one. Yeah. Uh, I should take a pen maybe. Yeah. So there was one experiment that one of my seniors actually performed where she was doing an EMG. So like EEG is a reading of brain waves, right? Electroencephalography. EMG is a reading of the nerve impulses that are sent to the muscle, right? So that is uh, uh, myography, if I'm not wrong, right? Instead of the brain or the cardiac in ECG, they are looking at the muscle stimulation, right? So it is called EMG. Over there also, there was this concept that I learned that, for example, there is a repeated stimulus, right? So if you are giving the same stimulus, I'm sorry, I'll just Yeah, I'm sorry. Uh, huh. So sorry for the kind of these drafts that I am able to draw here because that is how you can with a mouse, right? So my point is that, for example, you in that experiment, what happens is that there is suppose a muscle, right? And you have to understand that how the muscle reacts to a stimulus, right? So what you do is you open up, you cut a slit in the skin and you, or maybe in humans, that is also not required. Maybe you can just attach electrodes on the skin, and then what can you can do is you can give minor stimulation to that muscle, right? So minor electric stimulation. It is not like a shock. It is just so that you will know that the muscle is twitching or contracting. Those kinds of stimulation, right? So if you give multiple stimulations, right? At a particular time, right? So, for example, you have decided a time interval that is 10 milliseconds. So, after every 10 millisecond, you will give a stimulus and it will give this kind of an action potential to you, right? So all of them will overlap exactly the same, right? One, two, three, four, five, six, seven. So suppose seven stimulation you gave and seven such overlapping amplitudes you got, right? So this is a normal case, right? Based on this only, your a time interval for stimulation has been decided that is 10 milliseconds, right? Maybe if you give stimulation after every 5 milliseconds, so the muscle does not have time to relax after contracting, right? So it will not contract again, right? So then it will kind of be desensitized to that stimulus. So we know that 10 millisecond might be a good time, right? So this is one condition where you have given the stimulus. Now, why you were doing this experiment is you wanted to check whether there is some kind of muscle weakness or some kind of weakness of this signal stimulation that is happening in the peripheral muscles, right, in your subject. So, what you do is in your this was a control in which you had done like this, right? And then you take your experimental subject, and in that experimental subject, again, what you do is you give the same intensity or same stimulus at the same time interval, right? So for example, first it gave you such a peak, right? Thereafter again it gave you such a peak, right? But what you would see in that case, if this pathway is defective, is that this amplitude will keep decreasing, right? By the seventh one, maybe it has diminished like this, right? Instead of remaining a straight line, sorry, instead of remaining like a straight line over here, it has actually diminished in the second case, right? So this is a kind of thing that happens. So this signal intensity, what I mean to say is the stimulus intensity is essential, but that stimulus intensity is defined based on a normal condition, right? So it might be possible if you have some kind of a nerve impulse conduction issue or the sensory stimulation, in that pathway, if you have an issue, then your same amount of stimulus that should give such a response, this one, two, three, four, which should look something like this, might not look like this, right? That is a pathological condition there. Right, so these kinds of readings are essential in in finding out disease pathologies, also in basically diagnosing that, okay, now we know that some kind of muscle stimulation defect is there, right? So that is why we have actually kept these things in mind, or kept these things have been taught to you, right? Because these are the basic conditions, but these basic conditions are also checked in unhealthy individuals or individuals that have some kind of disease symptoms, so that we can find out which particular disease they might have, right? We can diagnose multiple such symptoms and come to a conclusion, right? Now, also, once you have a repeated stimulation, you like over there. we were just stimulating for like 7 times or 10 times, right? Which is a very, very nominal thing. But what if you have repeated, continuous repeated stimulation, right? For example, if somebody has gone to a tattoo parlor, for instance, right? Or somebody has yeah, put your feet in very cold water, right? So once you put your feet in very cold water, you will really feel chills, right? You will not want to get into that water. But if you sit with your feet inside that water, you slowly get to become accustomed to it, right? Your body gets accustomed to it. So it gets adapted, right? It is not that by putting your feet in the water temperature has changed drastically. No, that is not it, right? If another person comes and dips his or her feet in water at that same instant, the other person would also feel very chilly at that time, right? But that really doesn't mean that the temperature of water has changed. It is just that your receptors have become adapted to it or desensitized to it, right? So they have adapted to, okay, this is okay for my body. It is not really harming my body. Now I can get inside, right? So that is basically what is adaptation, right? So, for example, this adaptation is given in response to a touch receptor to different levels of touch, right? So, for example, if somebody, for how do I explain this? Um, again, yeah. I don't know if that is intentional or not, but if somebody is trying to speak up for the class, it is really not audible, right? Uh, yeah, in this case, slowly adapting a receptor. Okay, I'm just going to mute. Yeah, yeah. For example, you have something that is pricking your hand, right? Initially, it will take a moment for you to register that, okay, something has pricked me, right? But if somebody comes and pricks you again with that same thing, right? Maybe next time you respond to it quicker, right? And if somebody keeps pricking you for a while, your response will be much amplified, right? Or it much not amplified, your response will come much faster, right? Because now you are adapted to it. Okay, that somebody will come and will prick. Okay, now I like this much stimulus is what I required to actually feel it, right? So after a point, you will become desensitized to the first kind of prick, right? If it is not really harming you, you will get desensitized to it, right? Then later somebody will have to come and prick you harder or with at more points for you to realize that okay, this was pricking, right? Later they might have to increase continuously increase this threshold for you to actually respond to them, right? Yeah. And this is basically known as adaptation. So, if a stimulus persists for several seconds, the firing of neuron decreases gradually, right? That is why you don't feel very cold anymore inside that very chilly water that you dipped your feet in, right? Because your neuron, uh, it is called desensitization, but what is actually happening is that your neurons are not firing as much anymore, right? Yeah. So this generally happens in case of touch, it happens in case of smell, right? Even in case of smell, it happens, right? That once you enter a room and somebody has sprayed some room freshener in that room, you will automatically understand that, okay, there is this some kind of smell that is there in this room, right? And it is very, very catchy kind of, right? And you can get rid of that particular smell, but if you remain in that room for like 1 hour, you will not feel that anymore, right? Versus somebody who is just coming from outside will again feel that heat of smell, right? So you kind of become desensitized to it. So Yeah, these kinds of things are happening in case of both of most of the sensory neurons, right? Even in case of hearing, like if you are continuously hearing a very loud noise, after a while it probably might not disturb you that much, right? If you are continuously touching very hot water, after a while, provided that it is not burning you, after a while you might not feel it being as hot as you thought it was earlier, right? Similar such things are happening, right? But in case of visual stimulus, this never happens, right? So visual stimulus, we normally don't get adapted to or get desensitized that really, because because of this particular phenomena, which is known as microsaccades, right? So what are these microsaccades? They basically resemble regular saccades, which is regular small movements in most respects, except that they are very tiny and occur even when we are attempting to fixate the eyes on a particular location. For this reason, they are often referred to as fixational saccades. So, what does that mean? What does this term mean is that whenever you are fixating your eye on one particular object, right, even if you want it or not, you will assess various modalities within that object, right? And in between, your eyes might blink, right? Or involuntarily, you might look a little towards the side and then again at the back without you often realizing it yourself, right? So, because of these tiny changes in the eye movements that are, or tiny eye movements that are always taking place, your neurons are not, the same receptive field is not being illuminated for that long, right? It keeps on changing every microsecond. So that is why the neurons are not getting desensitized, right? For example, if I ask you to fixate on this particular face, right? And if you are looking at this particular face, you will also notice it's the woman's hair, the woman's nose, eyebrows, eyes, lips. What is the clothing that she's wearing? Like the dark circles beneath the eyes, the shadow that the photograph is giving, etc. Right? So this will be the kind of movements that will be recorded by your eyes. Right? You might realize these large saccadic movements, that is movements between you noticing her hair and her chin, right? Or her eyes and her clothes, or her eyes and her nose are still longer movements, right? Major movements. You might be able to realize that, okay, earlier I was looking over there, now I'm looking over here. But the smaller movements, like from looking at this portion of the eye to this portion to this portion to this portion, you will not be able to feel that your eyes are moving, but there will be micro level of changes that might be occurring, right? And your eyes are also obviously always blinking. So that is why they do not get the time to get desensitized. Yeah. So, from this, we know that visual perception indeed is a little different than the other kind of perceptions that we have been talking about, right? Over here, the eye movements are so sudden and so involuntary at times that there will be no fixation or adaptation that will occur over here, right? Also, you should note that the visual perception is more advanced than a simple camera, right? Also, we see more than the image that is formed on the retina, right? So, what does this mean? Because you have already gone through such lectures, you do know what is going to come next, right? So in a camera, what happens? Whatever you are seeing, it will be captured, right? Uh, in case of visual this thing, however, whatever you are seeing, you are also simultaneously interpreting, right? Because whatever you are seeing, that image is not formed in the eye itself, right? That information of what you are seeing is going to the brain. The brain is processing it in its processing center, and then it is sending it back. So while this processing is happening, the brain has not only recognized what this image is, it has also thought of why this image is over here, or if this image looks like this, why do you think it looks like this, right? So these are some very subconscious processing that has already been done by our brain, right? So for example, that is why it is written over here that we see more than the image that is formed on the retina, right? So ideally, the image that should have been formed is the inverted image of whatever I'm looking at, right? But I can see more than that, right? This phenomena is known as gestalt, right? So what is gestalt? It is basically a holistic perception, right? That is, we see objects rather than pixels. So if, for example, there were okay, the same example let's take, right? So if this was shown to me, what I would by default or instinctively, what I would see is two triangles that are overlain on top of one another and three circles, right, on the edges of one of the triangles, right? So this means that the brain will make assumptions about what is to be seen in the world based on experience and build in neural wiring for the vision, right? Similarly, if, for example, I don't know how many of you have texted before emoticons age had come, right? Okay, I will try to draw it nicely. If I draw this, right? People who do not know what this is might think that this is a less than sign and a 3, right? But before emoticons, how we used to converse is through text, right? And in text, you cannot add emoticons. So this would be a heart, right? Because this looks like a heart sideways, right? If you attach this, it looks like a heart. Similarly, if I draw a colon and a bracket, right? That is what I have drawn. I have drawn a colon and I have drawn a bracket. But what does it look like to a person who has used these kinds of representation? It does look like a smiley face, right? Similarly, a colon, where is my mouse? Yeah, a colon and a capital D, right? That looks like somebody who's laughing, right? So that is what is gestured. And that is what was actually used in these kinds of languages during texting, right? So now, although these are not very commonly used because of the presence of emoticons, obviously, but that is what it means, right? So our brain generally sees things in a holistic view, right? So it will not see individual pixels. Instead, it will see something in a more holistic manner. This example was again something that Sir had given. Right over here, it is very, very hard to make out what this particular image is of, right? It looks like something that is broken off and pieces are scattered on the ground, right? Or some, incomplete paint or just something that is torn apart, right? It looks something like that, right? But if you fill the gaps that are present over here, uh, maybe your brain might be able to make out what it is, right? So it looks like somebody spilled ink on top of the same pieces, right? The pieces have not been moved. Ink was spilled on top of each of them. Now, what does the brain see instead of these torn apart pieces? What does it see? It sees that there is a capital B written over here, right? Uh, which is what this processing does, right? So when one object appears to cover another, we assume that the occluded object is missing some of its part and fill in for it, right? So over here, we did not think that something was covering this, right? So we thought that it is torn pieces, but over here we think that something is covering this or occluding this yellow part. So we know that okay, this occluded part is B, capital letter B, right? Similar was the case of spares or cavities, wherein the decision depends, wherein the question actually asked was that whether these, what do you think, that these are hollow and or these are hollow, right? Which ones on the left or the right do you think is hollow, and which one do you think is protruded out, right? Is it like this or is it like this, right? So over here, most of us will think that this one is hollow, while this one is protruded, because the shadow is coming from here, right? The light is coming from here and shadow is forming over here. So that is what we might think. Right? Similar was this example that sir had given, another very nice example, that if you see over here, this is equal to this. But when this image is placed over here, behind in the corridor, it seems like these people are, seated in a corridor, and because the corridor is also getting narrow towards the end, and in the front we can see it as bigger, in the end we can view it as smaller, right? We assume that this particular lady is also sitting over here. It is not apart from another photo, right? But if we remove this particular clip from here and we place it right next to this figure, then what will we assume? That this figure is probably from another picture altogether, right? It has been cropped, it has been placed over here. We will not say that it was also present over here, or it was cropped and it was moved over here, right? That is not what we will generally say. So the perceived size of an object depends on assumptions about its position, right? So this is also a feature in the spatial resolution only that we were talking about. Similar is this object recognition, where edges are powerful cues for perception. So the outline drawing has clearly recognizable objects, right? Similarly, if for example, I were to draw something like this, right? So people outside of biology or outside of this particular course would consider it as clouds maybe in this setting. If, for example, this setting was not available and only this was drawn, right? You guys who are studying this neurobiology course would consider it as brain. Maybe it is, that is how line drawing of brain is generally drawn, right? If it is seen from the side, right? If it is seen from the top, it is somehow drawn like this, like there are two hemispheres. Okay, I don't think I will be able to justify even a line drawing with this mouse. So I'm just going to give it up. So, yeah, the visual information is processed at multiple levels in the brain, right? These are some of the levels that are given. You do not need to get into the, you know, this thing, the depth of it, right? This simple diagram that is given on the bottom is something that you can see, that there is a retina from which the optic nerve is coming. It is bringing it to the LGN, which is the region in the brain, which is taking to the V1 or the visual one sensory area. Half of it is going for processing in the dorsal parietal pathway. Half of it is going for processing in the ventral temporal pathway, right? And these various visual processing areas, V1 to V4, are being involved over here, right? So that is a simplistic view. Now, apart from that, there are other kinds of senses. Again, we were talking about visual sense for a while now. Now, there are other kinds of senses such as chemical senses, which are also called as chemoreception or chemosensation, right? What are the chemical senses that we generally come in contact with? They are either volatile chemicals, most of which are in the form of smells. Right? Volatile is something that will form fumes or come into the air, right? And you can perceive it from the air, right? So that is basically the smell. So the olfaction is one of the chemical senses, and the contact chemoreception, which is which is taste or gustation, right? So there again are multiple quadrants that we know of, right? we have talked about how odorants are working. They are taking it, what is a glomeruli in this case, how they are taking it to the cortical region, and then to that mushroom body or canyon body, etc. We have talked about it, right? Which is one of the key parts of this fourth, fifth module, right? Then there are some differences between vision and olfaction, right? Let us go through them also briefly. So visual input is low dimensional and can be described by a few parameters, right? These few parameters are color, RGB, that is red, green, blue. So whenever even you get a colored image, it is sometimes tagged as RGB. Whether a grayscale image is normally represented as maybe .jpeg, .jpg, but sometimes in some kinds of imaging, especially when you do it in the lab, the colored images are referred to as RGB, right? Or red, green, blue images. And intensity. Therefore, it needs only a few types of receptors. What are the few types of receptors that are present? Four types of photoreceptors, right? So these four types are the red, blue, green, four, three of those, and rods. Right? Just four types of photoreceptors are all right. But in olfaction, uh, uh, it cannot be described by a few parameters, right? Instead, it needs many receptors, right? So many different kinds of receptors are required over here. Then in vision, there is a lot of spatial information that we can get, right? Even if if I open my eye and all my other senses are blocked, right? If I am just seeing something, for example, if I am looking at a tab, right? Yeah, at my tablet, and I am looking at a video which is telling me direction from my hostel to my mess, for instance, from my hostel to maybe the auditorium of the institute, right? So what will happen is that I am understanding just through my vision how or where basically I need to go, right? There is a lot of spatial information that is involved, right? But in olfaction, it lacks spatial information, right? If I close my eyes and if I just smell, right? I might not be able to always detect where the smell is coming from, right? If there was just one kind of smell that exists, right? If I close my eyes and I basically smell something, and it smells like maybe rajma rice, or it smells like sambar jug, right? I might know that, okay, this is coming from the kitchen, right? That is the only spatial information I get out of it, right? But if there are multiple senses at play at one time, right? For example, you have gone to a party, there are multiple people have applied different kinds of colognes, right? Will you be able to make out which cologne smell is coming out of which person, right? It's very, very difficult. So there, it lacks spatial information. Vision. The visual input reaches cortex via thalamus, but olfaction bypasses the thalamus. This is also something that we have studied in the first module when we were actually going through these brain regions, right? Yeah. Olfaction is also used by a variety of animals. It is not just us. Even the insects have a very good olfactory system. So the olfactory system shares many features with the human olfactory system, and many lessons can be learned by performing experiments on simpler model organisms, right? Such as locust and honeybees and mosquitoes. A lot of odor related information that we have today is because of studies on these simple organisms, right? Drosophila also. So this is, for example, a setup. This is a locust, right? A grasshopper or locust, right? And these are the antenna. In most of the insects, antenna is the key organ for olfaction, right? Or which senses odor. So what has been done over here is that such a locust has been mounted and mounted such that it cannot move the rest of its body, and its antenna is being supplied with a particular odor, right? And then they are testing that what is happening to which and they have electrodes over here in the locus, right? So what they will test is that which neurons over here are firing in response to which odor, right? So that means that if I have 20 kinds of odors, I need to map that which neurons are firing when odor number 3 is being given, which neurons are firing when odor number 5 is being given, etc. Right? So that can be done. And that is how you understand olfaction and how different kinds of combinations can come together to basically aid this process of olfaction, right? So there is a combinatorial code at odorant receptors, which also we have gone through before, right? For example, if I'm talking about this first odorant, right, this molecule, it is very, very specific to one receptor. Only this purple colored receptor can, perceive it, right? So it is very specific to this receptor. On the other hand, this receptor might not just be very specific to this, right? This receptor with combination with this yellow and blue and navy blue, green and yellow receptors, it can basically detect this odorant, right? When it is in combination with this light blue, dark blue, yellow and lilac receptors, it can basically detect this odorant, right? So there is a combinatorial code that is playing a huge role in case of odorant receptors, right? And this is what it looks like. We have gone through this diagram also. So how is this olfactory response measured? There is this either an electrode is applied over here at this neuron at these neurons that are present in the antenna itself, or when the signal is taken from the antenna to this mushroom body or this group of Kenyan cells, there is an electrode that is applied on top of it such that there is a field potential that is captured, which is known as local field potential or LFP, right? It is a more summated response that is being captured, right? So that is how olfactory response is being measured, right? So over here, if we measured at single neurons, we will have sharp intracellular recordings. But if we measure at this region, at if we get a more summated response in a local field potential, it will be the average activity of many input neurons. Right, so it will not be sharp recordings like this, but it will be a summated activity, right? So, for example, uh, this is showing a response of a projection neuron or PN to different odors, right? So this is a projection neuron and this is an odorant that is LFP, right? Over here, if a cherry smell is given, the projection neuron peaks like this. If a floral smell is given, only a little peak is seen in the front and then it doesn't peak at all, right? When an apple smell is given, right, then again it doesn't peak at all. But when this cherry and apple smell is given, again we see it is not peaking at all, right? What does this mean is that when this odor, this projection neuron was only, receiving the smell of the cherry, right? It was able to respond. But when once this smell becomes mixed with the apple smell, either even if it is trying to respond, the negative response over here is canceling its response, and overall we are not seeing any response. Right? That could be one outcome or one way that it could be happening. The other way that it could actually be happening is that now once it is merged with this apple smell, it is no longer the same kind of smell, right? Individual apple smell and individual cherry smell might have been different. But once these two smells were merged, right, they are no longer the same smell anymore, right? That is why this projection neuron cannot go up because it is not the same smell. Also, there is a temporal pattern in the projection neurons, right? For example, if a projection okay, this is the same projection neuron that has been mapped, right? And the same odorant might have been given to it, right? So that means that once the order was given, it showed this kind of a response. Over here, the response was maximum, right? Again, the same smell was given again. That is why it is known as trial, right? So trial 1, 2, 3, 4 means the same smell has been given multiple times and similar kind of response has been observed over here, right? Now, this response has been given in a format that is more easily representable, so that it is known as raster format over here, right? It is what has come from the outcome of these trials, right? Now, let us look at the responses of a projection neuron with respect to 8 odors in the raster format, right? So over here, we know that the odor and this gray shaded gray area represents the time when the order is given, right? So this is the same projection neuron, right? So when order 1 is given, it is active, right? After that, this is the time when order is given. After that, it is activated. Order 2, it is not sensitive to order 2. It is not sensitive to order 3, not sensitive to order 4 or order 6, right? It is most sensitive to order number 5. As soon as the order is given, it starts responding and it shows a prolonged response, right? Then it is also a little sensitive to order 7 and a little sensitive to order 8, right? That is shown over here. Because these are activated or show a very high peak or spike in its activity after the order is given, right? Yeah. So the orders are encoded in the antennal lobe by identities of active projection neurons, by temporal firing patterns of projection neurons, and synchrony of projection neurons. Therefore, it is spatiotemporal code, right? So again, you don't need to get into details of that. What you need to remember is what kind of an outcome you get from what kind of measurement, right? So when you look at individual projection neurons, it is this kind of output that you get, the one that we were discussing in the previous slide, right? But what if you look at Kenyan cells, right? So Kenyan cell is where the actual final summated response is coming. So over here, although we saw that the projection neuron, this particular projection neuron that we were looking at was very, very sensitive to order number 5, right? But the summated response of all the projection neurons from the Kenyan cells or the mushroom body says that it will not respond to order 5 at all, right? Which orders is it responding to? 1, 2, maybe a little bit of 3 and 7, right? And 2 and 3, on the other hand, this particular projection neuron was not at all sensitive to, but it might be possible that the other ones were, right? So the summated response that is coming out is to be this, the one that is seen in the Kenyan cell. So the sparse representations may be easy to decode and associate with reward or punishment, right? So this is the over here, there is one other thing that you should note, that the projection neuron, it looks very busy, right? There are so many signals that are going on here and there. In case of Kenyan cell, if it is a summated response, how is it possible that the signal is so little, right? The signal looks very, very sparse, right? Why is it so little, right? Now that is because over here, you are just viewing what is happening when a stimulus is being performed, right? But there is just not this, information that is transported to the Kenyan cells. There is this information that is also transported to cells that are providing a negative feedback, right? So there will be some negative neurons or some GABAergic neurons in that case, right? Which will come and that will inhibit the response, some of the responses in the mushroom body, right? I think we see it in the next slide. So, for example, this is the response of a Kenyan cell to 5 orders. What is the mechanism of sparse responses in Kenyan cells is what we were discussing, and this I think you should be really aware of with respect to your exams also. That based on what we have seen in this course so far is that one possible reason that could be that the synaptic strengths between projection neurons and Kenyan cells may be weak. They are saying it could be sparse over here because the synaptic, association between these cells and these cells is weak, right? That could be one reason, but that is not actually the reason. even though there are lots of spikes in the projection neurons that are resulting in synaptic input to Kenyan cells, but the EPSPs that are produced may be small in magnitude. So you need lot of EPSPs to reach the threshold, and also the threshold in Kenyan cells may be relatively high. Now, this could be possible, right? We know that not all the cells will have same kind of threshold, right? Over here, the threshold for action potential may be higher, which is why, although the order can give these many responses over here, that response cannot generate an action potential over here, which is why this representation is this sparse, right? That could be a possibility. Also, there are intrinsic properties of Kenyan cells, but another possible factor that may be is that maybe Kenyan cells also receive inhibition from other neurons that keeps their activity in check, which is actually also what is happening, right? So this is the response of Kenyan cell to five orders, as we saw in the previous slide also. And then there is an inhibitory neurotransmitter, that is GABA, right? That we know has something to do with the sparse, signal, right? How do we know it is GABA? Because these authors in 2002, in the study that they have published in Science, what they had done is that they also suspected that inhibition could be playing a role in this, right? To confirm that, what they did was they blocked the inhibitory neurotransmitter using a drug that is known as picrotoxin. Right after this inhibitory neurotransmitter was actually blocked, what they saw was that there was an increase in signal in the Kenyan cells as well. So it means that on some level, this inhibitory neurotransmitter was what was playing a role in the sparse distribution of the signal in the Kenyan cell, right? So that could be one reason, but you can still see that this activity is still not as high as that in the projection neuron. So the other two reasons, that is lesser synaptic strength and more threshold in the Kenyan cells could account for those two, right? Also, there is maintenance of sparse responses by feedback inhibition. So there is a giant GABAergic neuron over here, right, with near the mushroom body, which provides inhibition proportional to the net activity of the Kenyan cells. So, depending on how active the Kenyan cells are, they will produce some response that will go and stimulate these GABAergic neurons, which will in turn go and, kind of inhibit it. So, it is a negative feedback that is happening over here. Also, this is what the vertebrate olfactory system looks like, right? So there is the olfactory epithelium that is present on the bridge of the nose. This olfactory epithelium is very close by to the brain, right? So it is basically separated by a cribriform plate from this olfactory bulb that directly connects to the piriform cortex inside the brain, right? Which is because of this, proximity of the nasal epithelium with the brain is why during COVID there were a lot of cases of brain swelling or some other neuro-related problem in patients that had severe COVID, right? Because COVID was going through the nose, it was affecting the nasal epithelium, and if it was severely infected, it could, it in some cases, it did transmit some of the infection into the across the cribriform plate into the olfactory bulb, and some neuroinflammation was also caused due to it, right? So that is the reason why. The structure of olfactory epithelium looks something like this, right? This is the electron micrograph structure. So each olfactory sensory neuron expresses one olfactory receptor. The types of olfactory receptors are around 315 in human and around 1,000 in mice, right? So mice can actually distinguish more smells than humans. Also, if we talk about dogs, right? Sorry, in humans, we have 5 to 6 million, but in dogs, right, the olfactory receptors are about 220 to 300 million, right? That is about 800 types of receptors. So they can distinguish those many smells. So that is why they have much stronger olfaction than humans. Then again, different odors activate different subsets of receptors, both in insects as well as in mammals, right? This was a discovery that actually won the 2004 Nobel Prize to Richard Alex and Linda Viva for the discoveries of odorant receptors and organization of olfactory system. Now, this also I want to point out to you guys that there were a few Nobel discoveries that you have learned within this course, right? So you should definitely remember them because they can be good questions in your exams as well, right? So, if we change the concentration of an odorant, it can also change the perceived odor, right? This is the concept which also is relative to if you smell too much of something, it might start smelling foul or toxic or stale, something like that. So, which means there is this one odorant molecule which is known as thioterpentol terpeniol, right? Thioterpeniol. Now, these particular chemical molecule, when it is present in low concentration, right? It smells like a tropical fruit. So, a tropical fruit might have this thioterpeniol and it smells like that, right? In a little higher concentration, it smells like a grapefruit, right? Grapefruit, again, it is a fruit. So, it will still smell citrus yet sweet, right? And it will smell like a grapefruit. But if the concentration of this particular compound is even higher, it will smell putrid, right? You will not be able to tolerate it smell. It will smell very, very bad, right? So it is the same odorant molecule, right? But depending on what concentration it is present in, the smell of that we are perceiving actually will also differ, right? Yeah. This is what the glomerular map looks like in case of mammals also, right? And in case of insects also, we had seen the same, that there are neurons that express the same odorant receptors. Now, these will basically gather an odor and they will send it from the olfactory epithelium to the olfactory bulb. And in the olfactory bulb, there will be glomeruli that are present, which will have a convergence of these neurons that is coming from the olfactory epithelium and also from top the neurons that are actually carrying this information forward. They make a junction at these individual glomeruli, which is known as glomerulus in this case. And yeah, over here. So this is an olfactory sensory neuron, and this is the glomerulus from which the mitral cells or the different kinds of other cells will take this information forward. Right, this mitral cell is coming over here. There are tufted cells in some of these, which will take this information forward to the olfactory cortex, where the pyramidal cells will actually take the carry this information forward. Right? Yeah. This is the olfactory pathway in mice. You can go through it later. Then we talk about pheromones, which are species-specific chemical messengers, right? There are some species that release chemical substances into their surroundings to influence behavior or psychology of other members of the same species. Now, this line is very important. We have gone through it also. So, they are affecting members of the same species, right? So, that musk ox odor that is attracting a lot of females to the musk ox is actually not going to help any human, right? Because it is a species specific pheromone. But alas, people are still selling that, right? So, one example is 11-cis Vaseline acetate. Released by male Drosophila, which attracts females, but it repels other males, right? There are also many, like in insects, so it is very, very common and very, very well established that pheromones are present. That is how they communicate within one another, right? Yeah. So that will be the end of it. Yeah, there was one other thing that I said I would talk about. I can just say that orally because I have not included it in my presentation. Is that a lot of the questions in the discussion session were pertaining to actually understanding like how despite potassium ions being more abundant within the cytosol, how the inner membrane or the cytosolic site is still negative overall, right? So the answer to that is one, that the membrane potential is negative. It is negative because of the ions that are present inside. Also, the membrane bilayer by itself, if you remember, you might have studied in the basics of biochemistry sometime maybe, that the membrane is made of a lipid bilayer. Like, right, this lipid bilayer have some phospholipids that are enriched on the outside of the lipid bilayer membrane, and some phospholipids that are enriched on the inner side or cytosolic side of the phospholipid bilayer. Right now, the phospholipids that are enriched on the inner membrane are mainly phosphatidylserine and phosphatidylinositol. These by themselves, the because of the side chains that are attached to them, also give a negative charge to the membrane itself. That is one thing, right? Secondly, we are saying that the potassium ions are comparatively more abundant inside, but again, there are sodium ions that are abundant outside, and there are much more sodium ions present outside than the amount of potassium ions present inside, right? So the outside is more positive. And because we have even those many potassium ions inside, there are a lot of other anions also that are present inside, right? So that is why, because of the presence of those anions, and because the first potassium is continuously trying to move out through the diffusion channels, etc., the inner side is still electronegative, right? It is more negative, right? And one basic difference you need to understand is that if we are saying the potassium is more inside, means that, okay, if there are 20 molecules inside, there are 5 molecules outside, right? But if we are saying that there is more sodium outside, means if there are maybe 5 molecules of sodium inside, then there are 100 molecules of sodium outside, right? So you are seeing the difference in the ratio of these, right? So there are much more sodium outside than potassium inside. So that is also one key reason. Yeah. So I will just discard these changes, and yeah. Just a moment, guys. Hello. Yes. So sorry for that interruption. Do you guys have any questions? That was the part I wanted to actually complete, and update it also with you guys, and in the notes as well. But if you guys have anything to ask, please do. I'll be I'm really sorry for the interruptions. So if you guys have anything to ask, please go ahead. Otherwise, we'll end the session, and all the best for your exams in that case. So I will take that as a no maybe, and we can end the session. First, let me just end the recording in that case.